Appreciate the few of you that are still in the room. I think you're not going to be sorry. My name is Dr. Kevin Slaywin. I am a physician. I did prostate cancer surgery for 25 years. In 2004, I started a company called Bellicum Pharmaceuticals. It was the first CAR T-cell company. 2014, I took it public at a $500 million market cap. And me and my team exited in 2017 and founded this company. And we've been working on it for the last 8 years, and I haven't really told anybody about it or what we're doing until this year. The CAR T-cell company was in cancer. This, of course, is in longevity. We think what we have now will be 10 times bigger than what we did before. I'll try and go through this very quickly, but if anybody wants my deck, just find me on LinkedIn and I'll send it to you. We've discovered a technology we call mitoxel geropeptides, and they target a fundamental cause of aging. In all aging organisms, the mitochondrial membrane potential goes down with age. This is due to something called proton leak. So your intermitochondrial membrane, it gets old, it gets leaky. The proton gradient across the intermitochondrial membrane, which produces ATP, just starts to get dissipated through the leak, and your mitochondrial efficiency, which typically when you're young, you'll make 34 ATP per molecule of glucose, but as you get older, you'll make 32, and then you'll make 30, and then you'll make 25, and then you'll make 20. And I don't know what the number is, but there is some number, and it's not compatible with life. So that's universal in all living organisms. We've discovered a technology that targets the inner mitochondrial membrane. Within 4 hours, the proton leak is completely eliminated, and your mitochondria go back to full efficiency, 34 ATP per molecule of glucose. So your mitochondria go back to when you were young in 4 hours. The other interesting thing about, and if you look at the phenotype of that correlates with improved exercise. If you test aging mice and you treat them, and then you put them on a treadmill or a rotarod, their exercise endurance goes up dramatically. And it doesn't go down ever again. It just goes back to 34 and it just stays there. So looks like aging is stopped. The other very interesting thing, same target, but we call it mechanism 2, is that if it's a senescent cell, it gets eliminated within about 8 weeks. So our technology will eliminate anywhere from 60 to 80% of all the senescent cells in the body within 8 weeks in every organ of the body, including the brain. So in summary, a single target, the inner mitochondrial membrane, and the reduction in the mitochondrial membrane potential that occurs in all living organisms of the age. The single target leads to 2 mechanisms, either immediate rejuvenation and restoration of the efficiency of your mitochondria back to when you were young, or if you're a senescent cell, elimination. So the technology is not binding any particular receptor. It's not like a GLP that binds the GLP receptor. It's based on the amino acid sequence and the physical properties. And so we have a certain set of rules that govern the sequence of these peptides. And so if it follows the certain rules, then you're a mitoxel peptide, it'll do these things. So we have a class of peptides, not just one, that does these things. So we're clinically developing 2 peptides right now, and our lead clinical candidate is 2105. But these class of peptides are anywhere from 18 to 30 amino acids in length. They cross the blood-brain barrier, so we eliminate all the senescent cells, glial senescent cells in the brain, they're gone. And we give it once a week, like a sub-Q injection, sort of like Ozempic or Mounjaro's given once a week. And we've typically been giving it, let's say, 8 to 16 weeks. Here's a hypothesis we had. With mechanism 2, if we eliminate the senescent cells and therefore reduce inflammation in the body, we should then see a phenotype from that. So let's see what happens. By the way, what I'm showing you now is 1% of our data. We've done over 20 well-controlled studies. We always use naturally aged mice, 75-week-old mice. It's not some fancy genetic model, they're just aging mice. So here's 75-week-old mice treated for 20 weeks with our peptides. We had 2107, 2105. You can see by any marker of senescent cells, senescence-associated beta-gal, P16, any of them, you'll see about a 60 to 70% reduction in the senescent cells. What happens when you get rid of these senescent cells? Well, the body all by itself, without doing anything else, starts to remodel itself back to when you were young. So for example, if you look in the muscle, as you get older, your muscle develops spaces between the muscle fibers. You see those white spaces? That's called intermyocyte space. The older your muscle gets, the bigger those spaces get. With treatment with our mitoxel technology, the intermyocyte space goes back to when you were younger, they disappear. Same thing with adipocyte area. The size of your fat cells gets bigger as you get older, but with our technology, they return back to a younger phenotype. This is pretty interesting, and I don't think anybody's ever seen anything like this. This is a dose response treating naturally aged animals with our peptides for 20 weeks. This is a dose response, so we're giving higher and higher doses, starting at 2.5 milligram per kilogram up to 50, and you see increasing loss of fat. You can see at 50, you get more than a 50% reduction in fat. The best you can do with a GLP-1 is maybe 15%. So it's 3 times better than a GLP-1. But what's remarkable is on the other side of the graph, which is in the same dose response, you see an increase in muscle. 15% increase in muscle, which is unprecedented. All the GLP-1s, every single one of them, for every pound of fat, you lose 2/3 is fat, but 1/3 is muscle. And I just want to make one very important point. We didn't target the fat, and we didn't target the muscle. We just targeted the aging defect in the mitochondria. This all happens all by itself. This is kind of a nice time course, 30 weeks with saline control, the gray line. These are naturally aged mice given a high-fat diet. You can see the gray saline, they gain about 25% of their weight over time. That's typical. Look at semaglutide, the pink one. They lose weight initially, but it starts to wear off. I'm a physician. When you're on semaglutide, Ozempic, it works at first, but then it starts to wear off, and you got to get on higher and higher doses. It's well known. Now look at 2105. That's the blue circles. In the first 8 weeks, the line overlaps with saline because it takes 8 weeks to get rid of your senescent cells. And that's what's causing the remodeling. So in the first 8 weeks, you don't see anything. But then you can see that the blue line starts to go down. And by 30 weeks, you're doing better than semaglutide. Okay. And then if you add 2105 to semaglutide, you can see in the first 8 weeks, it overlaps to pink. But after 8 weeks, you see it does better than semaglutide alone. And at 30 weeks, you're 14% more weight loss. With mechanism 1, rejuvenation of the mitochondria, look at the blue circles on the left. That's the optimal dose, 25. Within the first 5 weeks, the mice exercise 25% better than before. These were old mice, and now they're exercising as if they were a lot younger. And notice that that line just goes straight. And as long as we follow them, it's just straight. You would expect if they were aging that it would slowly go down again, but it doesn't. Because the mitochondria are at full efficiency and they stay there. So one theory is that we've arrested aging. Here's a really interesting experiment. I did the same exact experiment in 20-week-old mice or in 75-week-old mice. Everything else was the same. In the 20-week-old mice, our technology doesn't do any differently than saline in weight loss. They don't lose weight because they're young and they don't need to lose weight. And we're not targeting the fat, we're targeting aging. So on the left, 20-week-old mice, they don't lose weight. But 75-week-old mice, they lose weight because they need to lose weight. So it shows we're targeting aging, not fat. What's also interesting, here's a dose response. So 2.5 and then 25, you see more and more weight loss. But we were wondering, is this like a GLP-1? You lose your appetite or you're sick. You know, GLP-1s, you have bloating, nausea, vomiting, you don't eat. And then you lose weight. So we were wondering, is that's what's going on here? So we actually measured the food intake. You can see aggregate consumption, they actually eat more. It's like going to McDonald's and losing weight. It'd be nice. I'm almost done. Last slide. I started this experiment almost a year ago. I was like, all right, well, people have showed that if you increase the mitochondrial memory potential, it'll extend life in animals. They've also shown that if you get rid of senescent cells, it can extend life. Well, we do both. So we're like, all right, well, let's see if we can extend life. So a year ago, I started this experiment, and it's just starting to get interesting. I'm just going to stand up here for a minute. This is saline. So this is, let's say, cancer-specific. These mice are starting to die of old age. And if you look at this curve, that curve is going to look like this over the next few months. So those mice are dying of old age. Notice the 2105 and 2107 are not. They haven't started dying yet. Now, I don't know when they're going to start dying or if they're going to start dying, but I'll let you know over the next 3 months. But I just want you to know that already reached. Another mouse died today. I didn't get a chance to fix it. Another saline mice died, and the p-value is 0.05. So I'm up here showing it because it's significant. How much more it's significant will get, I don't know. So if you just look at fat mass on Ozempic, Mounjaro, all of them, 11% loss of fat, 16% loss of fat. Ours is 34%. And then if you look at muscle, 4% loss of muscle, 6% loss of muscle, 23% gain. All right, we're done.